majoring in biology. I'm from Dr. Matthew Gentry's lab in the Molecular and Cellular Biochemistry department, and I'm excited to present my research to you today, personalized medicine for fatal epilepsy, Lafora disease. Lafora disease is a devastating genetic form of epilepsy that manifests during childhood. These children experience increasingly severe seizures while also suffering from symptoms of cognitive decline and difficulty controlling their muscles. Typically, they will die in their early 20s. So what drives Lafora disease? If you could direct your attention to the bottom left image, you'll see a brain tissue slide with an arrow pointing to a dark fuchsia circle. This is a Lafora body, which is an aggregation of sugar that kills neurons and therefore drives disease progression. And while all patients with Lafora disease exhibit similar symptoms, the age of onset and the rate of progression varies significantly from patient to patient. And because of this variability, Lafora disease patients are good candidates for personalized medicine. So today, I will walk you through my project of how I characterize mutations within Lafora disease in order to aid the development of creating personalized therapies. So first, I will introduce you to the protein laforin. Then, I will walk you through a pipeline developed in order to characterize laforin mutants. And finally, I will share my results. Laforin is a protein that has the critical function of regulating how our brains store sugar, especially in the brain. As you can see, laforin is made up of two domains. And patients with Lafora disease often exhibit DNA mutations that lead to the production of laforin mutants. But to understand how that could lead to Lafora disease, let me discuss two key functions within laforin concerning its domains. The blue domain binds to sugar, while the domain in purple is responsible for removing phosphate from sugar. If there's an impairment in either of these functions, Lafora bodies form. And this makes sense because we know that laforin is responsible for regulating sugar storage. And if that's impaired, Lafora bodies would form. Our lab has developed a pipeline with a series of tests in order to characterize these mutations. So we can take novel mutations, run them through the pipeline, and then based on the characterizations we draw, we can classify them into groups in terms of their severity, such as group A, B, and C, as demonstrated by the top right image, showing the spectrum of Lafora disease severity in terms of the groups. But what does this pipeline look like? I will demonstrate to you what it actually looks like by walking you through how I used it to characterize three recently identified patient mutations: G24R, which occurs in the domain in blue, A268T, which occurs near the active site of the domain in purple, and V249L, which occurs in another part of the domain in purple. So the first step of the pipeline is to test for the functioning of the domain in blue. And if you'll remember, that's the ability to bind to sugar. And this is measured in percent sugar binding ability. So the greater interactions and ability the protein has to bind to sugar, the higher the percent sugar binding ability. And as you can see, when we compare this to the wild type, the G24R mutation has a significantly reduced ability to bind to sugar, and therefore we can characterize this mutation as having an impairment in the functioning of the domain in blue. The next step of the pipeline is to test for the functioning of the domain in purple, and if you'll recall, that's the ability to remove phosphate. So therefore, we measure this in phosphate released. And when we compare it to the wild type, once again, you can see a mutant is having a significantly worse time in performing near wild type levels, and this is A268T. So therefore, we can characterize this mutant as having an impairment in the functioning of the domain in purple. And when we look back at the data in terms of how the laforin domains are performing when we compare it to the functioning of the wild type, V249L seems to be doing a good job. However, the last step of the pipeline is to look at the overall stability of the protein, and we measure this in melting temperature. And as you can see, the V249L mutation has a significantly reduced melting temperature when compared to that of the wild type, which is actually close to our body temperature. And that has severe implications for how it would function in our bodies. So as you can see, these three mutations had three vastly different impacts on laforin's functionings. And that can be used to explain that variability that we see in the age of onset and the rate of progression. And as we discuss these mutations, we can't forget that these are three individuals that are suffering uniquely from a disease without a cure. However, with continued characterization and research, we can build the foundation for developing personalized therapies that can improve the quality of life for these individuals and their families. Thank you for your time, and I will now be taking questions.
I have a question. Can you explain what a wild type is?
Okay, so the wild type is the, sorry, you're fine. Wild type is the unmutated form of the protein.
So meaning as it appears in the wild, so to speak?
Yes,
I got
what
it.
it's supposed to look like.
Okay, yeah, a term of art I haven't heard. And are there any known therapies to treat this disease?
So currently, what physicians are doing is they're just addressing these symptoms and treating them, and they'll change the diet of the patient to like a keto diet in ways to kind of reduce the severity of it, but currently no. But excitingly, the parts of the Gentry Lab are currently working on therapies for this disease, with the first, one of them going to trial in 2021.
So I've got a couple of questions, mostly just my ignorance. But the first is, on these charts, the the bottom axis, WT, I'm assuming, stands for wild type.
Yes.
Do the others, which I recognize correspond with the above and the 3 individuals, but they are not correlated with groups A, B, and C at the top right. Is that correct?
That is correct.
Okay. Now this is the ignorant question. I know what phosphate is. I think I know what makes up sugar, but I'm not sure I do. And for you to say, here's a slide of a brain, and that's sugar, I mean, I love sweet tarts, but I'm guessing it's not quite the same circumstance. So what is sugar here, yeah, chemically, and what's going on?
So the sugar that we're looking at in Lafora bodies, it's actually aggregation of glycogen, which is made up of glucose. So that's what we're looking at in that image. That's what you're seeing. And the phosphate that we talk about, the reason that's important is because if we're not able to remove the phosphate from the sugars, then we can't break it down and get rid of it. And so it aggregates and it builds up and it affects the functioning of neurons.
So can I ask one more question on that? So your title is Personalized Medicine for Fatal Epilepsy. You said at the very end that this research is helping us to potentially do that, but what is it? What would the personalized medicine be? Take us beyond just this slide.
Okay, so as I mentioned, the Gentry Labs is currently collaborating with 2 companies to create 2 independent personalized treatments. So this can be applied to that, in that once we characterize each mutation, we can classify them into different groups based on their severity. And so clinicians in the drug trial, they can use this and they can identify which patients would be the most optimal to receive the treatment. So in this instance, it would be group C, because the Laforan that is less severe, those patients that have mutations that are less severe in Laforan, would be better candidates and more likely to respond to the treatment. And then furthermore, when addressing like groups A and B, when physicians have this information and know just how serious their patient's disease is and what kind of prognosis they can expect, they can then alter their treatment plan to like increase dosage and just be more aggressive overall.
Thank you very much.
Thank you.
This has been really a fascinating talk, and even though I'm not a scientist, I'm pretty sure I followed what you were trying to explain to us. My question is, what kind of human tissue sample are you working with for this kind of research? Is it a, you know, are you working from blood samples? Are you working from who knows what? Can you explain to me what goes into doing this research?
Yes, so I'm actually, parts of our lab are concerned with addressing the tissues, but I just grow the protein itself in the lab and experiment on it then. But like that brain tissue slide, that's a brain tissue slide that you're seeing over there. But that's basically where we're seeing Lafora bodies mostly, in that those aggregation of Lafora bodies in the neurons is the most critical. So if you were to look for tissues to study, I would suggest the brain. Thank you.
Any other questions? Okay, thank you very much.
Thank you for your time.